Hello, everyone. Welcome to our breakfast seminar. My name is Max Self. I work in the R&D department at Diagnosis. Here's an overview of today's seminar. So I will just briefly talk about our software products and everything we have to offer at ASMS this year. Then Mario will talk about his project in high-throughput phosphoproteomics, where he's mapping the signaling networks in yeast that depend on phosphorylations. And after that, Birgit will give us insights into protein acylation and dietary effects on succinylation specifically. So, as I said, I will give a brief introduction to the company. So Diagnosis has been around for about 13 years now. We have a highly qualified workforce, and a lot of publications are based on our products. We also have one of the largest facilities in the world for large-scale discovery proteomics, about 10,000 square feet or 900 square meters. We have customers around the world, and we have three main software products. Spectronaut, which is the software that you will hear a little more about today, as well as Spectral for targeted proteomics and Spectromine for data-dependent acquisition, specifically isobaric labeling quantification. And just a note on Spectromine, there's a new version of it coming out later this month, so stay tuned for that as well. Our software in general is characterized by high performance. It's also very well accepted by the scientific community. And one of the most important things for us is to make it easy to use. So you have a lot of analysis steps integrated into the software, and everything is as easy to use as possible. And we work with our customers to improve our products continuously. So before we get started, a reminder on data-dependent acquisition versus data-independent acquisition. So in data-dependent acquisition, individual precursor peptides get selected for fragmentation in MS2. And this gives you very clean MS2 data, but of course, you can't get fragmentation data for all precursors. And in the end, you have incomplete data for all the precursors across the retention time range. On the other hand, in data-independent acquisition, many precursors get fragmented together at the same time. This produces very complex MS2 data, but it also gives you fragmentation data for all your precursors across the entire retention time range. And typically, you would analyze DRA data with a library that you generate with data-dependent acquisition methods. But this is something that we have started to challenge a little bit. So you can generate high-quality libraries from DDA data in Spectronaut, but you don't have to. You can also just use the direct DRA workflow that allows you to essentially analyze your DRA data with DRA data only without acquiring any additional files to make a library. As we will hear, Spectronaut is great to look at PTMs. Now a few words about Diagnosis at ASMS. We submitted a lot of abstracts for the scientific posters and talks as well. So we did get 3 scientific talks. So later today, Norman will talk about how we improve Spectronaut with machine learning. Luca, in the afternoon, will present a high-throughput immunopeptidomics pipeline. And on Wednesday, I will introduce how deep learning scores are improving our poster search engine. We have 10 posters in total. And you're very welcome to visit us at booth 224, where we will give you all the information you might like about our software, as well as our services. Next Wednesday, we have another breakfast seminar. So if you like the breakfast, there's more of that. in 2 days. And yeah, we will have a presentation about the past, present, and future of Spectronaut by our CTO, Lucas Reiter. And Brett Finney will show how he uses Spectronaut in a proteomics core facility. Any information you might want is available at biognosys.com/ASMS2021. So yeah, you can just go to that website, find all the information about the posters and talks. And with that, I would like to introduce Mario Leutert. Like Biognosys, he is from Switzerland, where he did his undergrad and got his PhD in Zurich. And then he moved to Judit Villen's lab at the University of Washington, where he's working on phosphorylation-dependent signaling and high-throughput proteomics. So with that, Mario, please take it away.
All right. Yep, thanks a lot for the kind introduction, and thanks to Biognosys to give me the opportunity to present my work here about the high-dimensional atlas of phosphorylation-dependent signaling in budding yeast. So I'll give you first a short overview over a method that we have developed called R2P2, Rapid Robotic Phosphoproteomics, which allows us to do really large-scale phosphoproteomics. I'll then share you I will talk about the large-scale, high-dimensional perturbation-phosphoroproteomic experiment where we use DIA mass spectrometry together with Spectronaut in budding yeast. And then I'll share some insights into the architecture of the yeast signaling network that we gain by this analysis. Okay, so first, as a small introduction, we need high-dimensional measurements which are required to define signaling networks. So you can imagine in the case of phosphorylation, nowadays, with mass spectrometry, we can get really deep coverage of the phosphoproteome. However, the difficulty is to really make sense of the phosphorylation sites and relate them to each other. So most of the times you're interested, you have a perturbation of your cellular system, and you want to know what are the main responses. But for this, you need to be able to connect the important phosphorylation sites. You need to be able to define what is crosstalk with other pathways, what are complementary responses, and in the case of phosphorylation, there might also be promiscuous phosphorylation. So the way to do this and to bring some hierarchy and structure into the phosphoproteome, one thing we could do is do a high-resolution time course of a perturbation, or we can apply many different perturbations to the cellular system and thereby make some sense. What are the connections between the different phosphorylation events? So, however, this is not trivial in phosphoproteomics because phosphoproteomics is limited in throughput and reproducibility. The reason for this, for one, is that you have many sample preparation steps that need to be performed before you end up with your phosphopeptides. And then also on the analytical side, there's difficulties because for phosphoproteomics, you rely on peptide level readouts, and you also need to be able to localize the phosphorylation within the peptide sequence. So our approach to tackle this is we developed our R2P2 method, which you can do in a 96-well format. It's streamlined and automated. And then for the downstream analytical analysis, we use DIA mass spectrometry, data-independent acquisition, in order to reproducibly sample our phosphoproteomes and also gain the quantitative accuracy. So in the past, there have been several papers that basically show that all of these upstream sample preparation steps are doable using magnetic bead technology. So we took these concepts and combined them in a single workflow, which we implemented on a Kingfisher Flex magnetic particle processing robot. So this robot takes 96 well plate and is able to move magnetic beads between the different plates. So that's how our method looks like. It comes in 2 robotic runs. So first R2P2. It takes cell lysates in a 96 well plate. Magnetic beads are moved into the cell lysate and mixed together with high organic solvents. So proteins precipitate on the magnetic beads. And then the magnetic bead protein complexes are moved through a couple of different wash plates, and thereby you purify your proteins and desalt the sample. We then do an on-beat digestion, and after drying the samples down, they're ready for mass spectrometry analysis. However, you can also take the peptides and subject them to a second robotic run, and there we use iron IMAC beads to enrich for phosphorylated peptides, and again, move them through different wash plates, elute the phosphopeptides, and again, after drying them down, the phosphopeptides are ready for mass spectrometry analysis. We've shown in the past that this method, R2P2, shows really good selectivity, so no matter the protein input amount, we end up with really highly pure phosphopeptides, so usually more than 98% of the peptides are actually phosphorylated. And we also get good quantitative reproducibility. So high identification rates within and across different batches, and the batch here would be different 96 well plates or different robotic runs. And we also reach good quantitative accuracy within and between batches here, judged by Pearson correlation between the phosphoproteomes. Okay, so this is now published. You can have a look. It's in Molecular Systems Biology, and we have quite a lot of details in the methods in there if you want to do this yourself in your lab. Okay, so now we have this method in hand, and we're basically ready to really perform high-dimensional phosphoproteomics. And so we do this in budding yeast. So budding yeast is a great organism to do this. First of all, it has many conserved pathways and obviously also reduced genome complexity. So it's really good for like big systems biology type of studies. We chose to, and another reason is that budding yeast is a single cell organism. So it has, and it needs to be able to live in many different environments. So it has a lot of signaling pathways in place to sense internal as well as external perturbations and rapidly adjust to them. So in this experiment, what we did, we exposed yeast to a very diverse set of many different perturbations, so 106 different perturbations. And then importantly, we only applied this perturbation for 5 minutes and then directly harvested our yeast cells. So that means that's kind of the time span where the phosphoproteome is really changing. However, the underlying proteome, as well as gene expression and so on, is not yet changing. We did this in 6 biological replicates and ended up with around 600 to 700 samples, which we distributed across 796. So we distributed the lysates across 796 well plates. And then the actual proteomic experiment basically came in 2 branches. So first of all, we took a pooled sample, which contained all of the perturbed samples. We did an R2P2, then very deep offline HPLC fractionation using a pentafluorophenyl column or a strong cation exchange column, and then acquired around 100 data-dependent acquisition MS runs. And so this gave us a really deep yeast phosphoproteome. Then on the other hand, we also processed all samples or all plates individually. So we run them through 7 R2P2s, and then acquired like 600 to 700 data-independent acquisition mass spectrometry runs, which gave us our quantitative phosphoproteome. And so now we use Spectronaut to create a hybrid spectral library using these DDA runs up here, as well as some of the DIA runs that we acquired down here. And we also use Spectronaut again to perform a spectral library search on our quantitative phosphoproteome. I will mention a little bit more to the Spectronaut approach later, but so I thought for this seminar it's also interesting to show you how we set up our experiment, since you can imagine for such a big experiment it's very important how you distribute your samples and how you create your batches. So here a batch is basically one robotic run, right? And you need to feed in 196 well plate. So on each 96 well plate we have our perturbations scrambled. However, importantly, we only have always one biological replicate of each perturbation on a 96 well plate. So the replicates are distributed across the different 96 well plates. This, as you will see, is important to omit possible effects, possible batch effects. Also on each plate we had at least 4 pooled lysate controls, so they come from the same lysate and will afterwards help us to determine how efficient our digest and phospho-enrichments were in each batch, and also control for possible batch effects. So all these plates were run through R2P2, and then we acquired our quantitative phosphoproteome using an Orbitrap Exploris using a 60-minute gradient. And in terms how the samples were measured afterwards, so we have our sample preparation controls in the beginning that we measured to assess if everything went well. And then we had different blocks of samples, which again corresponded to the actual batches that we prepared up here. And then in between the different blocks and within the blocks, we always had controls, which was just a pooled phosphoproteome, and all of these were then acquired in exactly the same DIA method. And since it's such a big experiment, so between the blocks, they're not necessarily run consecutively, but there might have been other experiments in between. And that's kind of our pipeline with Spectronaut. So we have our DIA-MS raw files. As mentioned before, we then perform our spectral library. And here, importantly, I think that's a really big benefit of using Spectronaut. We use the pipeline approach within Spectronaut that allows us basically to process batches of samples as we go and immediately do QC in real time, basically, as we measure. And then in the end, we can combine all of the different sample batches that we've already done our spectral library searches on, apply global FDRs and localizations. Then in the end, we basically export two tables from Spectronaut, so the precursor data, which we mainly use for QC purposes, and also the aggregated phosphorylation site table, which I think is a new feature of Spectronaut. And that's then the table we do all our downstream bioinformatic analysis on. That's just an example how our QC would look like. So from all the parameters we can, or all the measurement parameters we can export from Spectronaut. So maybe just like, so basically here, the y-axis or the x-axis are our individual mass spec runs in chronological order. And then, for example, we can have a look at MS2 points per peak, or also at MS2 precursor intensity, which is all logged here. And then down here, we track the different LC-MS parameters, for example, sample batch, but also whenever we have to change the analytical column and calibration, and also bake out and even cleaning cycles on the mass spec itself. And so that really helped us to weed out samples that had, like, for example, bad chromatography, but which we didn't catch immediately. And also, I mean, you can do like really long-term QC, and you see, for example, here, kind of get fewer points per peak, and that's most probably related to the column exchange down here. But also, like, intensity gets better in the end, and that's because we just did a quadrupole cleaning of the instrument. And so this is kind of the global structure of the phosphoproteomic data. So over the whole experiment, we measured something like 37,000 phosphorylation sites. Here we plot the median intensity of each phosphorylation site and the proportion of samples which have a detection for this phosphorylation site. And obviously, this is one of the biggest challenges in phosphoproteomics here, so you will have a lot of missing data because it's a peptide level measurement, basically. But if you look here, so samples that have, or phosphorylation sites that have been measured in most of the samples, they're usually the high abundant sites, and then you have this logistic distribution, and obviously sites with lower intensity in general, you also measure them fewer times, and that just shows that the missingness in the data is not at random. We apply quite a strict filter that we only consider phosphorylation sites that have been seen in at least 50% of all the samples. Don't talk about this here, but we also saw, obviously, I mean, if you have a big experiment like this is quite likely. So we saw some batch effects, which corresponded to the different robotic runs. And so we were able to take care of this quite easily by first doing an imputation correct with combat batch correction, and then remove the imputed values. And that's basically the final corrected data set we ended up with and did our analysis on. And so yeah, in this data frame, we have really good identifications of around 5,000 phosphorylation sites, which are reproducibly measured across all perturbations. And then also replicate correlation looks good. with most of the samples having a Pearson correlation above 0.8, and then some which look quite bad, but that's actually related to the perturbation itself. So there are some really harsh perturbations, and they just have a much more stochastic phosphorylation response. Already with like this kind of simple methods, here again, Pearson correlation and hierarchical clustering, we're now able to pull apart the different perturbations from each other by perturbation type. So in this dendrogram, you, for example, see here the perturbations which are most distant from everything else are pH shifts, also oxidative stresses, and then, for example, over here you have changes in carbon and nitrogen sources, osmotic stresses, and then down here it's more like smaller clusters together of, for example, DNA damage agents or proteostasis perturbations. Yeah, however, this data is really complex, so we also need a little bit more advanced data analysis methods to look at it. And something that worked really well for us is dimensionality reduction by UMAP. So basically here what you do is you perform a normal principal component analysis. You take your 50 or 100 principal components which carry most of the variance, and then perform a UMAP analysis to bring it to the same two-dimensional space. And so here each dot would be an individual phosphoproteome of an individual perturbation or individual sample. And you see we already get quite some separation and clustering. And if we color it by sample preparation batch, we see that it's not due to sample preparation batch effects. But if we color by perturbation type, we see it's actually the perturbations which pull the samples apart. And again, over here you have carbon and nitrogen source variations, pH stresses, and you also start seeing like interesting clusters of different perturbation types. So for example, down here, osmotic stress condition, heat shock, and cell wall perturbations. Still have, I mean, there's like this bigger part here. There's still separation within, but these are perturbations which are have more like specific responses, not super broad responses. So now this is a really great analysis to look how are the different perturbations related to each other, how is like the phosphoproteome changing in general, but what we are most interested in is how are the different phosphorylation sites related to each other. So we can do exactly the same analysis, but in this time each point here would basically be an individual phosphorylation site, and then get at how are these related to each other. So we did this, and then we looked for phosphorylation sites which are co-regulated. So that means if you have over the whole perturbation landscape, are they going up in a similar way or down, or how do they look like? So we did this, and then we grouped phosphorylation sites which are regulated in a very similar manner into modules. So a module would, for example, be a group of 30 phosphorylation sites, and they show very similar responses across the whole perturbation landscape. And so if I walk you really quickly through this heat map, so here each row will be an individual phosphorylation site module. So that means these modules contain anywhere from 20 to 90 phosphorylation sites, and then each column will be a specific perturbation. And obviously, so for example, if you look at this cluster here with these modules, you see, oh yeah, phosphorylation sites in this module are really highly upregulated in starvation conditions, but they seem to be very much downregulated in proteostasis conditions. And we really like this kind of modular type of analysis, but yeah, as you know, most biological systems are somewhat modular in a way, and if you now think about phosphorylation site modules, what could it mean? So it could be, for example, sites that are all modified by the same kinase, could be sites that are in a similar pathway, or it could be sites even on the same protein complex. So now what we can start is like, yeah, make sense of these phosphorylation site modules. You can just perform normal enrichment analysis, compare it to other data sets. Here I compare it to kinase interaction data set. And basically, that's shown here in this heat map. So again, each row will be a module, and then each column will be a specific yeast kinase. And so, for example, here what it means is in this module, you have kinase or interactions of that kinase enriched. So that's likely corresponding to kind of some signaling hub or some hub where this kinase modifies a lot of sites and shows specific regulation in one of the perturbations. So let's zoom in here where I marked. And we can look at 3 modules specifically. So for example, you have this module down here. And most of the phosphorylation sites, or the phosphorylation sites in this module, they are usually not regulated or down-regulated, but then you have a few conditions where they are highly upregulated. And that's, for example, in this case, for osmotic stress conditions. And then, not surprisingly, in there we find a kinase hub which is enriched, consisting of 3 MAPK kinases, which are all involved in the high osmolarity MAPK signaling pathway. We can also look at these 2 modules up here. They're quite similar to each other. And they are highly enriched in SH9 and TOR1 kinase interaction partners. And we see they are usually constitutively phosphorylated, but then in a few conditions, like the starvation condition, you lose phosphorylation in this module. And yeah, we can now look. So this module is consists of around 54 phosphorylation sites. So now we can really go at the highest resolution level and look at the individual phosphorylation sites. And we see that around half of these phosphorylation sites in this module are located on proteins which are already known to be in the TOR signaling pathway. So for example, here on the catalytic subunit of TORC1 itself, and then also on 4 downstream kinases of the TORC1 pathway. So we have SH9, and here indicated in red, these phosphorylation sites were actually already previously known to serve an activating function of SH9, and they're known to be phosphorylated by TORC1. But then you also have here the really downstream transcription factors, which mediate basically the effects of activation of this pathway. Then in the same module, we have these 2 kinases, which are also nutrient-responsive kinases, but don't have a known role in the TOR1 pathway, so there might be crosstalk to other pathway or previously unidentified signaling. And then obviously we have half of the proteins where the phosphorylation is not known what it does, and it's also not known how it is related to this pathway. So these are now the interesting ones that we want to look at and see how can we make sense of this. And to circle back completely, we can now take this module and map its expression on our perturbation UMAP that I showed in the beginning, which is shown over here. So aggregated intensity of these phosphorylation sites. And indeed, like the module is constitutively phosphorylated in most of the conditions, and then we have a few clusters over here where phosphorylation is lost in this module, and this will be again the starvation conditions, which make sense for TOR1, and the high osmolarity conditions. Okay, so as a summary, we performed this really large-scale signaling experiment, and now I showed you that we can generate basically a map of co-regulated phosphorylation sites and get conclusions out of this, but we can also predict kinase activity landscapes, and something we are doing together with the group of Pedro Beltrao. We can take this really large-scale data and try now to predict which phosphorylation sites are actually functional. And all of this basically gives us insight into how the signaling network works. Okay, so with this I'm at the end. Thanks to Judit Villen for having me in her lab, and also Anthony, who did a lot of the computational work. And so if you want to know more about the project, I have a poster today. We have another phosphoproteomic paper poster today, more from the computational side, and then a couple of talks and presentations.
Thank you very much, Mario.
Okay.
We will take questions after Birgit's talk. So please, if you have questions, write them down or don't forget them. There will be an opportunity to ask. But in the meantime, I think we need to make the switch here for the microphones. And in the meantime, I can introduce our next speaker. So it's Birgit Schilling. She's an associate professor at the Buck Institute, where she's also the director of the Proteomics co-facility. She has been developing innovative analytical technologies for a long time now, and she's involved in a very wide range of proteomics projects now. She's a longtime user of Spectronaut, and she will talk about using direct DIA for looking at succinylation in different contexts. So yeah, I'm really looking forward to her talk, and yeah, we're very lucky to have her here at our breakfast seminar today.
Thank you, Max. All right. Thank you, everybody. I'm so happy to be here. This is our first conference in person, so I'm really happy to start this off with the diagnosis workshop. And thank you, Max, and everybody. So yeah, thank you so much. And PTMs for breakfast, that's a fun title, and we really embrace this. As Max said, I have my own lab at the Buck Institute. We're just north of San Francisco, and so there you can see we have a lot of interest in neurodegenerative diseases, but also I'm very involved in biomarker work and sarcopenia, proteostasis, and also biology of aging in general. So today I will talk to you about PTMs in the context of a lot of metabolic disorders. So, thank you, Mario, for talking a bit about the phosphorylation. That's like really a key chemical modification. In general, the PTM space is huge, right? So, I mean, there are really many PTMs, very complex, ubiquitination, even proteolysis. I like to present to you a little bit today about acylation. This is really important in signaling and many diseases. So let me show you a little bit about that. So, but in general, what are the challenges of PTM? So they are diverse. Often they're low site stoichiometry, so we often have to enrich them. So, and also it's really important, what is the site? The localization is key, and of course, there's crosstalk. Mario already mentioned that also. So, it's really important to localize. And also, PTMs are really, by definition, one-peptide wonders, right? So you need to get the quantification right. There's only one shot, basically, right? And so we're using direct DIA. And thank you, Max. You introduced this quite nicely before. So let me show you a little bit strategies in DIA, right? DIA is kind of complex, and you can do it many different ways. Maybe in the beginning, a lot of people, including us, would actually do DDA acquisitions and then build your own spectral library and then use that in order to quantify the DIA acquisitions. Or you could take just DIA, just do the DIA acquisitions, and either use existing libraries or build in silico, and that's where the direct DIA comes in. Or you can build spectral libraries from the DIA directly, such as DIA umpire, or do some fancy chromatogram libraries. So we actually, most of in the beginning we did DDA libraries, but now we totally switched to direct DIA. And for PTM work, it's actually really nice because it saves a lot of material. So when we enrich, we don't have that much. And so we save material and time, and it's very accurate. So we really like this workflow. So let me show you some examples. So we have some sirtuin knockouts. Sirtuins are involved in longevity and very important in aging. They're HDAC histone deacetylases, right? They're involved in chromatin formation, so they're very relevant. And 35 is actually a D-succinylase and a D-malonylase. So it means it takes off the succinyl group or the malonyl group. And if you have a knockout, you would expect that those levels are increased from those modifications. So we do enrichments with antibodies from CST in parallel for succinyl, malonyl, and then we do a little bit the protein level, and this is all Jacob's work. He works hard on this. This is not easy, and so I appreciate that. And then nowadays we run everything through our Orbitrap Eclipse. And I think the direct DIA works really well because it's so good mass accuracy, and Joanna is running those. And and then using direct DIA and Spectronaut for everything. So how would you set this up in Spectronaut? So normally, if you have a DDA library, you would select this kind of workflow up there. The direct DIA, there's another kind of entry level, and it's really easy to use. So we actually really appreciate that. We have so much data, so we can run this through Spectronaut really quickly. And then there's actually a PTM workflow module here, and we set PTM localization. We set multiplicity, and so it's a very easy workflow to do for PTMs, which is highly appreciated because, as Mario said, right, we're not at his level of automation, but still we have quite a lot of samples. And then you just select your modification of choice here for us, succinylation, and then everything else is pretty much default as Biognosys has it in their default parameters. I like to point out for us, we set the Q value sparse. So sometimes in the sirtuin knockouts, it's really high levels, and in the wild type, it's nothing. So we don't want to, so we need to also get some reading in the wild type, so Q value sparse works. And we do not want to normalize, right? That's the biology right there. So we check no for cross-normalization. So then, we really like the IRT, calibration. That works great. It's also a nice QC. Does everything work fine in all the samples, or maybe we lost something here? And then, I like this new PTM plot. It actually shows you here, the screen bar is the succinylation. So you can already see wild type knockout. There's more numbers of this modification. But of course, in addition, we're even more interested in the high-fold changes, of course. So, but these kind of plots you get right away out of Spectronaut, and we like looking at those. And, it's actually interesting when I was telling Max before, when you do your big experiment and you open the Spectronaut first, you open the volcano plot and it's either nothing or your experiment has worked, right? So we love, it's kind of, I'm always a little nervous to open this, but you can kind of see on the fly. So in this case here, we had the 35 knockout over wild type, and you can see these succinylated peptides, they're all massively upregulated. And I'd like to point out also, I think this is like 20 log 2, so it's a huge fold change, right? So it's really a massive reorganization. You can right away look at some heat maps, right? This is all available in the data analysis feature in Spectronaut. And also we like the PCA analysis. In our case, these knockouts really clustered away very nicely from the wild type. And you could imagine, just having your breakfast and just looking in Spectronaut real quick, did the experiment work or not? So anyway, maybe I do this, or maybe I let Rana do this. And okay, good. So let's see. So, there's a PTM, there's actually a nice PTM site report nowadays. There's a localization filter that is available. We check that. It's easy to do. And then you get all the information from that. It's also interesting, when you get so many results, you cannot check everything. But for the things that are important, we like to check and look at the data a little bit before we tell our collaborator to follow up on something. So that's really easy to do in Spectronaut these days. So in fact, what I really like are these features. You can, so for example, in this case, this is an acetylation experiment from before. You could have the site either on this K or on that K. And, you can simulate in Spectronaut, okay, how would it be if the K would be here? And you get confirming ions and then refuting ions and certain scores. So for example, one of the first confirming ions would be this Y2, confirming that the acetylation site is on this lysine. And you can see when you click on this, it marks for you which transition that is. And it's really nice and very, very confident, in fact. And Spectronaut also, simulates the other isomer, and that had no confirming ion score. So if you want to, you can look back at the raw data, and we really do this a lot indeed. And, we appreciate that we are able to do this. And, the other thing I wanted to point out, there is, for example, in the PTM site report, The site probability can be seen. You get a score there. You can filter by that score. You can be very stringent. We usually are at 0.75, and I saw Mario had that same filter, but you can be more stringent, And I mean, here it's all very close to 1 in our case. You can, for example, have number of PTMs. You see multiple peptides, maybe different charge states, or you can see certain things that have PTMs on multiple PTMs in one peptide. So it's really all very easy to see. You can also, on the fly, do some gene ontology. For you to see a little bit easier here, I highlight a few. For example, in this experiment with the 35 over wild type succinylation, you see a lot of metabolic pathways that were enriched, and that was important. Maybe showing you a few specific experiments. So we saw over 2,000 succinilated peptides and around 600 malonylated, and of those that were significant, you can see an overlap of 48. And we kind of like that. That's like a PTM crosstalk, right? So the site could be either malonylated or succinilated. So what are those proteins and peptides? So I'm showing you a few here, and I'd like to point out, see these log2 ratios of like 18, right? I mean, in normal space, that's 200,000-fold upregulated. So what that really means, it's like it's there in the sirtuin knockout and not at all in the wild type, right? So I think the PTM space is really, it's like black and white sometimes, right? It's on and off, and it's very dynamic. So I really love that about PTMs, right? That's really signaling in a cell. And yeah, let's see maybe how does this actually look when you look at these sites. And in Spectronaut, you're able to look at the traces and XICs. So yeah, you see in the wild type, there's really nothing. In the sirtuin knockout brain, very nice signal. And you can look at the different transitions, and the malonylated peptide on the same site also looks really nice. So we really appreciate that it's not a black box, right? We can look at the data and really convince ourselves this is really true. And I think this is really important. I mean, there's biology behind these things, so that's really what we're interested in. So what is the biology, right? So so here you can see some volcano plots that Joanna made, and a lot of brain-related proteins. I mean, this is from mouse brain. I wanted to point out again, most people look at PTMs in liver, and we do too, but in the brain it was important. So we saw some nice upregulation in, for example, Parkinson's involved in Parkinson's disease, synuclein or tau here, just to point out a few. So, what is their relevance in aging and neurodegenerative diseases? As I said, the sirtuins have some longevity aspect in them. And, but for example, with the synuclein, we were interested to look at that closer, and we just plugged it back into Skyline. As many of you know, I like Skyline. So, we can just switch back and forth between the different softwares, and you can see really nothing in the wild type and really strong signal again in the sirtuin knockout. So really a strong phenotype, but why do we do all this, right? So a few years earlier, we actually looked at acetylation in the context of AD and cognitive decline, and we found several acetylation sites, and we followed those up and made like mutants for certain acetylation sites on tau. So, and they really had to do a lot with synaptic plasticity and memory loss. So what our collaborator did, and I hope you can see this, this is like a Western blot for tau and of the hippocampus of a mouse. And then we found certain acetylation sites in tau itself, and our collaborator made some mimetics, mutations of K to Q. It mimics acetylation genetically. And you can see that when you do the tau mutation on the endogenous level, you see already a lot more tau aggregates. And when it's a little bit overexpressed for this mutant, you see massive amounts of aggregates. So we really try and do some biological follow-up on some of these events, and that's why it's so important to be really confident in the existence of the PTMs. So, what do you do with PTMs? Why are they relevant? And I just wanted to show you one extra example. We're really interested in biomarkers, using PTMs for biomarkers. But let me show you in the last 3-4 slides a study that we did, and which is really interesting. And so, we have a collaborator in Pittsburgh, close by, right? A couple of hours. And they have some kind of diet. It's just a nutritional supplement, something you would eat anyway. And when you give this to some wild-type mice, they are healthy, and then, actually enrich for succinylated peptides, what would you get? Can this diet somewhat improve health of these mice? So let's see what happens. And, so apparently, we just recently did a little bit some work with Bruker and using their DIA-PASEF workflow. The other work I presented earlier was done on the Eclipse, and this is now done on the Bruker-Timstorff, having the extra ion mobility space to reduce complexity. Anything reducing complexity is good. So, and then we basically run all this again through Spectronaut in the workflows I showed you before. And what we see, what I really wanted to point out here is actually, if you, for example, in the report here, the post-analysis, if you filter for succinyl here, you can see all your candidates. And you can click on any peptide here and see, for example, how does this peptide look and the site actually in the control diet versus the nutritional diet. So I just wanted to emphasize again, the easy, accessible visualizations. But when you have this filter on, it's really interesting when you then look at the volcano plot. Well, I mean, we were quite surprised about this volcano plot, right? I mean, with this just simple diet, all of a sudden there's a massive regulation in both directions, up and down, in these mice, right? And when we looked at the protein level, there were like 5 proteins changed or something, right? Maybe 20 proteins. So on the protein level, nothing happened. So if somebody does just proteomics on the protein level, they say, well, this diet does nothing. But when you look at the PTM, well, it's a massive regulation. And actually what I wanted to point out is only the red dots are only the succinylation sites. There are a couple of gray dots. Those are non-modified peptides. So you can easily, because we set that in the candidate list like this, so it's really nice. What was interesting is the ones that go up are mostly peroxisomal. The ones go down that are mostly mitochondrial. I'll get back to that in a second. And actually what is really nice when you hover with your cursor over something, it will tell you, oh, this is this peptide with that modification on that protein. So it's very interactive, so you can mine it very quickly. As I said, if you get up in the morning, first thing you do, you just have a quick look what you have. Or you could even, say, somebody's interested in this particular peptide here that comes from some ACAT protein. You can type it in the custom selection and then just see, oh, where is it? And then it will highlight those in the volcano plot. So it's very easy to initially visualize, and then of course we do all our bioinformatic pipeline downstream. But initially you can get a very nice impression of what your data is, if one has to redo a run or acquisition, or maybe just use, I don't know, just get some initial results to the collaborator. Anyway, why is this so exciting? I don't know. This is really cool. This nutritional supplement in the liver that I'm showing you here totally shifted the metabolic pathway from fatty acid oxidation that normally happens in the mitochondria over to peroxisomes. So it's a total remodeling, and in fact, this really gives good energy. It generates acetate and succinate, and then that in the end can go through the complex 2 and electron transport chain and generate energy. Why is this important? So there's a lot of metabolic disorders in mitochondria. For example, and in these mice that were wild type, it improved treadmill performance and in general metabolic parameters. In some of the mitochondrial diseases, in fact, in these mice, when you give them the diet, they increase runtime, blood ketones and blood lactates get lower. In a talk that I give on Wednesday, I will show this particular diet in a kidney injury model, and there it also really helps. But the mitochondrial, so for example, a lot of metabolic diseases are really bad for kids and, people die early. So this helps them improve their metabolism. So we really try and make this a translational aspect for humans. But in general, well, I hope I could show you Spectronaut is a key factor in our lab to run our experiments. We use DIA Direct, and we like the visualization tools. We think PTMs can be biomarkers. I think that's really something my lab wants to go to. That's really one thing that I think is a little bit understudied, and Mario showed nicely how relevant this is also. And, as I showed to you a little bit, some of these diets we think can rescue liver function and protect against acute kidney injury. And for us, we like the quantitative accuracy. I think it's key, and also it's key to have good QC and to be confident in the data. Anyway, this is my lab. Several of my lab members are here. We're so thrilled to be in person at ASMS, and I like to point out Jacob and Joanna and everybody in my lab. And thank you, Max, and everybody for having me here today. Okay.
Thank you very much, Birgit, for this very nice talk. And with that, we'll open it to questions. So Mario, if you could just come up here so I can hand my microphone over. So if you have questions, please walk up to the microphone so that we can capture it for the recording as well. So if you, Lucas has a question, so let's start with that.
Thank you both, very nice presentations. I have a question. You said that you think many PTMs are black and white, and we obviously see the same, so that's very interesting. At the same time, it's challenging for data analysis, particularly statistics. So can you elaborate a little bit whether you have been playing around with imputation strategies and how this interacts with the statistics performed, and it would be very interesting to hear.
Well, you're right, it's a challenge, right? Because often in the wild type, there's just nothing. So we have to really see that we integrate something. And we have used the Q-value sparse in order to do this. And that has been quite nice. We also check that it then actually integrates in the correct space. But with the DI Direct, it has a predicted kind of retention time and then would integrate there. We also tested a few outside software packages, and often we cross-check them. But we think in Spectronaut, the algorithms, the defaults are quite nice there. And that has worked for us. But you're right, that's a definite challenge. And for DDA, LFQ, for example, that would be really difficult, And that's why I think the DIA is so nice for this type of workflow. Mm-hmm. Thank you.
Thank you both. Very interesting talks. I had a question about the software that obviously I'm not familiar with this or with high throughput proteomics, so that was very enlightening. But can you utilize this to filter through ion mobility as well? Because I know you mentioned that at one point, but I wasn't clear on that.
Utilized for what? Can you?
I'm sorry?
Oh, can you repeat the last part?
Oh, I was asking specifically for ion mobility. Are you able to use this software to filter not just by phosphorylation or mass to charge, but additionally by ion mobility?
Oh, yes.
By collisional cross-section?
Yes, so the report, in fact, gives you the ion mobility factors, and we actually looked a bit at the data, right, and we were interested if there's PTM isomers that have different ion mobility. So the reports export all that, and then, so that's actually a nice way, right? So it reduces the complexity, and yes, one can look at that. That's nice. Thank you.
Thanks. I have a question for Mario. So can you give us an idea of how much time it takes to do the sample prep versus sample or data acquisition versus data analysis for these large-scale projects?
Yeah, the sample preparation on itself is really fast. It's more the sample generation. So in my case, exposing the yeast to all the stresses and then the lysis. The sample preparation is really fast. Then the analysis is also really fast, especially with the spectral library approach. So that's fast. And then, the bioinformatic analysis, you can spend years if you want.
Yeah, I also have a question for Mario. In your phosphodata, how often do you see that phosphosites on the same side of the protein are co-regulated? And or how often do you see patches on the surface of proteins? Do you often map it to the 3D structure of the protein and look at the data like that? Or because nowadays you get a lot of structural information through AlphaFold, for instance, and it's widely available.
Yeah. I mean, that's definitely like an avenue we go a lot in the lab, and it's specifically interesting for phosphorylation because here we see that the average yeast phosphoprotein or the average yeast protein has around 6 phosphorylation sites, and then, for example, kinases can be crazy with up to 100 different phosphorylation sites. So yeah, it's really interesting to observe where the hotspots are and how could they be in protein-protein interfaces and so on. Yeah.
Mm-hmm. Yes. I mean, actually, I would like to say, yeah, it's really interesting. Sometimes we have these hotspot proteins that have so many acetylation or succinylation sites, right, that seem to be really key regulators. And in our case, a lot of them are involved in the peroxisomes. And that's the fun, right, looking at the biology afterwards and trying to make sense of that. And that's where the close collaboration with collaborators come in. And that's where the fun starts, right?
I have a quick question for Birgit as well. So this nutritional supplement,
yes.
I guess I'm just wondering what it might look like. So in peroxisomes, there's alpha oxidation. So is it reasonable to assume that this might be a beta-branched fatty acid?
Yeah. I think you're in the right direction. Yeah, okay. I think it's a fatty acid derivative. So what the nice thing about that is that it's something that we would eat anyway, right? So it's all FDA approved, and we actually did some work even in monkeys, you know. So it's amazing. It improves the metabolic, mitochondrial, and peroxisomal function in the liver, and in kidney it actually protects against injury, which is so important, right? Because in chemotherapy, a lot of people have kidney problems or, cisplatin, and also ischemia in kidney happens. So you could imagine, just taking the supplement for a while or eating some coconut, and and then after a while just stopping that again, right? So that's one of the nice things about what therapeutic interventions, right? You also want them to not linger on after you abandon them. So we're very excited, yeah. And this comes out of Pennsylvania, there in Pittsburgh, my collaborators.
Yeah. I think we have to close it here. Is there any other question? We would maybe have time for one more. But otherwise, we will have to leave the room in the next 5 minutes. So thanks everyone for coming. Please remember to visit our website, diagnosis.com/ASMS2021 for all the information about what we have to offer at the conference. And please also consider coming back for the breakfast seminar on Wednesday. Thank you very much. And thanks to the speakers for their great talks.
Thank you.